Hey, Hammy here, back with video 3, in which we're going to look at if there is no oxygen, how can the cell continue to get some ATP energy through the process of fermentation. When we talk about the process of fermentation, remember that we ended up in glycolysis, splitting glucose, glycolysis, into two 3-carbon pyruvates. Okay, now those 3-carbon pyruvates are what they call a pivotal metabolite. Okay, depending the fate of that pyruvate depends whether there is oxygen available to the cell or not. If it is available, down here, if oxygen is available to the cell, the pyruvate goes into the mitochondria, and the complete aerobic breakdown or cell respiration of that pyruvate, of that organic molecule, happens, and we get the most ATP out of it. If O2 is not available, the cell still needs energy in anaerobic conditions. So there are processes they go through called fermentation, different types of fermentation, which glucose is incompletely broken down, but we still get, remember, you can still go through glycolysis, which gives you a net gain of 2 ATP. Okay, so it allows still some energy production to continue. On this slide, we just see an overview of the process of fermentation. So the first part right here, okay, is going to be glycolysis. Okay, so here's our glucose. Remember, we have to invest some energy. We put some 2 ATP in, goes to G3P, to BPG, we get out some electrons. We fill up NADH with electrons, E negative. And then BPG to pyruvate, we get our 4 ATPs, which is our net gain of 2. The problem is that the cell can keep going through glycolysis and gaining 2 ATPs, so it's getting a little bit of energy even though there's not oxygen present. The problem is this process can only happen if you have, that's supposed to be NAD plus there, if you have free electron dump trucks freed up to pick up the electrons. If there's no NAD plus here, okay, that prevents a roadblock, and glycolysis will slow down and stop. Okay, and so you're not going to get this gain of 2 ATP. We have to, as cells, they have to have somewhere to put those electrons from NADH to offload them to free up NAD plus to come back so it can keep going through glycolysis. Okay, so what it does, what cells do in fermentation is they offload those electrons into the pyruvate molecule. The 2 most common types are lactic acid fermentation, this route over here, where the electrons are put into the pyruvate and it is reduced into lactic acid. Or the other most common one, alcoholic fermentation. The electrons are put into the pyruvate. Oftentimes the CO2 is split off, and you get 2 ethyl alcohols or ethanols. Okay, so it's just a way, fermentation is a way of changing that pyruvate, putting the electrons in, freeing up the NAD plus to come back up here and allowing glycolysis to continue to happen so the cells can continue to get energy in the absence of oxygen. This is just the overview, inputs, outputs slide showing you, again, you're starting with glucose, and it's going to be broken into 2 pyruvates, which then, depending on the type of fermentation that's happening, will either be made into 2 lactic acids or 2 ethyl alcohols and 2 CO2s. And again, here's the goal. You're going to get a +2 ATP gain. So without oxygen, the cell is still able to get ATP energy. So just a reminder, fermentation is an anaerobic process, okay, that reduces pyruvate either into lactic acid or alcohol or some other things you'll see here in a second, okay, which frees up the NADH back to NAD plus and allows glycolysis to continue. Of the major types, alcoholic fermentation carried out by yeast make our alcoholic beverages, spirits, also breads. But most of the alcohol it's a very low amount, and when you bake the bread, the alcohol evaporates. The second type, lactic acid fermentation, a lot of us are familiar with, happens in our muscles, and it all, can kind of make us sore. But it's also used to make things like cheese, yogurt, sauerkraut. Other bacteria, there's other types of fermentation you'll see here in a second to make, other types of commercial products. This is a slide I threw in here just showing you some of the different, depending on the type of organism you're using for the fermentation, some of the different products that you might be familiar with. Okay, this organism here is going through fermentation, making CO2 and propionic acid, making Swiss cheese. Okay, the CO2 bubbles make the holes, the little gas bubble holes in the Swiss cheese. There's different bacteria here making lactic acid, our cheddar cheese, yogurt, soy sauce. Okay, soy sauce is actually fermented. Here's our ethanol, our alcoholic beverages, the wines, beers, those kind of things. There are other types of alcoholic fermentations, isopropanol, which would be like rubbing alcohol, acetone, nail polish remover, acetic acid fermentation, how we get the vinegar. Okay. These are all different kind of subtypes of fermentation. Advantages of fermentation. It can happen very quickly. It's just glycolysis. It can run through those 10 steps really quick, and you're getting ATP fairly quickly. This happens in our muscles when we do rapid bursts of some type of strenuous exercise, weightlifting, golf swing, bat swing, sprinting, those kind of things. Disadvantage, the lactic acid can build up if we do it too much, cause muscles to fatigue, to cramp up, not letting the muscle fibers relax and causing a cramp, which causes oxygen debt, which some of you know, and then you're sore for a few days until that lactic acid is carried out of the muscles in the blood back to the liver, where it's made back into glucose or glycogen. Alcohol that is produced can actually be toxic to the cells. A lot of alcoholic spirits only get to about 5% to about 16%. And then the alcohol that the yeast are producing will actually kill them. To get more concentrated spirits or higher alcohol content spirits, that's where you have to do the distillation. You have to boil the alcohol off, collect it, and try to concentrate it. The efficiency, you're only making 2 ATPs from the glycolysis, which is about 14.6 kilocalories. Whereas if you completely broke down glucose if you had oxygen, that's 686 kilocalories. Okay, so that's only an efficiency of 2.1%. So not real efficient, but it's, you're only 2 ATPs instead of 36 or 38 with doing complete oxidative breakdown, but it's quick. It can happen without oxygen in anaerobic conditions, and so there are advantages. There's times where this is helpful. All right, that sort of wraps up fermentation. In the next video, we're going to be looking at what happens if there is oxygen and the pyruvic acids can be taken into the mitochondria and you get complete oxidative breakdown of that pyruvate.